These are the recorded lectures for Slide Set, Chapter 3, Part A. There are actually 4 parts. There's A, B, C, and D. So this is a really long chapter, but it is one of the review chapters, and the information, you may see the information on the test that you take to get into allied health programs. The TEAS test is the one that most community college nursing programs use. So Chapter 3 is on cells, and Part A, I'm going to skip. Part A is going to start with the cell theory, and then we will cover the basic structure of cells. So the cell theory basically says all living things are composed of cells. The cell is the structural and functional unit of life. And I should go back to my first statement. The cell theory says that all living organisms are composed of at least one cell, because we know that bacteria and some other microorganisms are unicellular or single-celled, but humans are multicellular. So cells can only arise from other pre-existing cells, from the division of other cells. This page, I don't have anything highlighted, but it's interesting to note that there are over 250 different types of human cells, and this is just a very small sampling of those. These cells that you see in the top left are fibroblasts. They're found in connective tissue, and they produce protein fibers like collagen and elastic fibers and reticular fibers. Erythrocytes are red blood cells. Epithelial cells are, these are actually cuboidal epithelial cells, and because they're shaped like a cube, but they are found in linings and coverings, like the skin is epithelial tissue. This is a skeletal muscle cell, which is multinucleate. That means all these little purple ovals are the nucleus. So this muscle cell has many, many nuclei rather than just one. Erythrocytes, or red blood cells, are anucleate, meaning they don't have a nucleus at all. Smooth muscle cells have a single central nucleus. Smooth muscle cells are found lining your blood vessels and your digestive tract and your airways. This is a macrophage, which is a very large white blood cell that engulfs anything that needs to be broken down. So it can engulf bacteria and break down and destroy bacteria. It can engulf old, worn-out cells and break them down and recycle them. This is a fat cell, which is completely filled with fat. The nucleus is shoved off to one side because the fat fills up the cell. This is a nerve cell. This is a sperm cell. So lots of diversity in human cells. But all human cells have 3 basic parts. All human cells have a plasma membrane, which is the outer covering of the cell. It's composed primarily of a double layer of phospholipids with proteins and cholesterol scattered throughout the phospholipid bilayer. The cytoplasm is everything inside the plasma membrane, and within the cytoplasm there are organelles or cell parts that have certain roles. They do certain jobs inside the cell. The nucleus is the largest of these organelles, and it stores the DNA. It is the control center of the cell. Now, this is an image of a cell, a typical generalized cell, and the cell organelles that compose it. So you need to be able to label this. Okay, so this large purple structure is the nucleus. The entire structure is the nucleus. The pores that you can see here, they are called the nuclear pores. I don't think they're labeled. They're not. Okay, so there's a nuclear envelope. That's a double membrane. There are pores called nuclear pores that allow for molecules to leave and enter the nucleus. The DNA in the nucleus of a cell at rest is all scattered and spread out, and it's called chromatin. And then inside the nucleus, you can have one or more of these structures. This is a nucleolus, but sometimes there can be two, sometimes more. The nucleolus, so the role of the nucleus itself is to store the DNA. The role of the nucleolus is to produce ribosomes. And ribosomes are these little red dots you see. Some of them are scattered and some of them are attached to this membrane, this channel of membranes called the rough ER, or rough endoplasmic reticulum. So we have the rough endoplasmic reticulum, which is attached to the nucleus, to the nuclear envelope. We have the smooth endoplasmic reticulum, which is attached to the rough. And then we have the Golgi apparatus. We have ribosomes that can be free in the cytoplasm or attached to the rough ER. We have the cytoplasm, which is everything inside of the plasma membrane. We have the mitochondrion, which produces ATP, lysosome, which functions in digestion, and then we see just a typical vesicle here. It's not labeled, but it contains products produced by the Golgi that are being secreted outside the cell. We see protein fibers or filaments. The smallest ones are the microfilaments. I don't see any of those labeled on here, but then the medium-sized ones are intermediate filaments, and then the thickest ones are microtubules. These structures are called centrioles, and the centrioles, together with their spindle fibers or microtubules that function in cell division, they are called the centrosome. This vesicle is called a peroxisome, and it functions in breaking down fatty acids. So we divide the fluids inside and outside the cell. So if it is a fluid that's found outside the cell, we call it extracellular fluid, ECF. Extracellular fluid is also known as body fluids. The fluid that surrounds all of our cells is called interstitial fluid or tissue fluid. The fluid that surrounds our blood, our red blood cells and white blood cells, is called plasma. And then fluid that surrounds our nervous system, brain and spinal cord, is called CSF or cerebrospinal fluid. ICF is the fluid inside the cell, sometimes called the cytosol, intracellular fluid or ICF. And ECF is the fluid outside the cell called extracellular fluid. Now, section 2 is specifically on the plasma membrane and transport across the plasma membrane. The plasma membrane is composed primarily of a bilayer or double layer of phospholipids. And then there are membrane proteins that float in this phospholipid bilayer. And so we refer to a model of the plasma membrane as a fluid mosaic model. There are carbohydrate chains that are attached either to the phospholipids or to the membrane proteins, and they form the glyco portion of the glycocalyx. So glyco just means like a chain of glucose molecules. And then cells are attached to each other by cell junctions. So this is an image of the plasma membrane that I'm going to blow up. And you can see the phospholipid bilayer. You can see the heads of the phospholipids, which are called the phosphate heads, face inside and outside the cell, and then the fatty acid tails, which are insoluble in water, they face each other. So these are phospholipids. They contain a phosphate head that is soluble in water, and then 2 fatty acid tails attached. So this double layer of phospholipids has cholesterol molecules, which are these golden ring, 4-ring structures. The cholesterol helps to give the membrane more stability. And these large, chunky purple molecules are the membrane proteins, and they have various functions. This one is a channel protein that allows for transport into and out of the cell. And then you have these chains of glucose, these carbohydrate chains. This one would be attached to a protein, so we would call it a glycoprotein. This one is attached to a phospholipid, so we call it a glycolipid. But they're part of the glycocalyx. And here we see the functions of the plasma membrane are as a physical barrier. The plasma membrane allows selective permeability. It selects which molecules can enter and leave the cell. The plasma membrane functions in communication. It can interact with chemical messengers like hormones and enzymes. And the plasma membrane functions in cell recognition. The different types of glycoproteins and glycolipids on the surface will identify this cell as one of your regular body cells or self cells, or it will identify it as something foreign that your immune system needs to attack. And this page really goes over everything I just said, so you can look at that on your own. And the animation on membrane structure, I encourage you to watch this, is slide 14. Slide 14 of set 3A. All right, for the lipids that make up the membrane, the primary lipid component is phospholipids. Phospholipids make up 75% of the lipid component of the plasma membrane. Then there are glycolipids and cholesterol. And cholesterol helps to increase the stability of the membrane. And this just shows you a picture of an individual phospholipid has a polar hydrophilic head. Hydrophilic means it's soluble in water or water-loving because it's going to face the outside or the inside of the cell, which is probably surrounded by some type of fluid. And then you have nonpolar hydrophobic tails, 2 of those. Those are fatty acid tails that are water-fearing or insoluble in water. Then we have a section on the different membrane proteins, and I'm going to blow this page up too. There are 6 different types of membrane proteins. There are transport proteins, which either have a channel through them, we call them channel proteins, or they allow molecules that are going to be transported. They allow those molecules to bind to them. It causes a change in the shape of the protein, and it moves the molecule across, transports the molecule across. So this is a carrier protein. So transport proteins can be channel proteins or carrier proteins. Then we have proteins in the membrane that are receptors for hormones and other chemical messengers. So once that chemical messenger binds to that receptor, a change happens. Then we have enzymes, and those enzymes just catalyze specific chemical reactions across the plasma membrane. We have glycoproteins, which function in cell recognition or tissue recognition. We look at those glycoproteins to determine what the blood type is for red blood cells, and to determine which tissues would work for which organs and tissues would work for a tissue match in organ transplants and tissue transplants. We have the proteins that function in attaching the cell to the extracellular matrix, the substances outside the cells. And then we have junction proteins that join cells to other cells. Okay, so these are the different types of proteins that you find in the plasma membrane. There is an animation on them in slide 21 if you want to watch it. Then there's an animation on enzymes. This section is on the glycocalyx. Just remember that the glycocalyx consists of chains of simple sugars, carbohydrate chains that are attached to the lipid portion of the plasma membrane or the proteins, and they're always on the surface, not on the inside portion. So on the surface of the cell, you only find glycolipids and glycoproteins on the surface. These are important in letting the immune system know which type of cell it is. And then here's the last slide that we're going to go over in this recording. This is our clinical homeostatic imbalance, example 3.1. The glycocalyx of some cancer cells can change so rapidly that the immune system can't recognize the cell as being damaged. And therefore, that cell is not destroyed, and it's able to keep dividing and form into a tumor. And this is slide number 26. So we'll start the next recording with slide